Hi all, the presentation is going to be on saliva and secretion, salivary secretion. Introduction. The oral cavity is a moist environment. Saliva is a complex fluid. Human saliva consists of organic and inorganic components. The enzyme found essential in beginning the process of digestion of dietary starches and fat. Saliva circulating in the mouth is termed as whole saliva. Saliva reflects the physiological status of the body's mirror. According to Stedman Dictionary, saliva is a clear, tasteless, odorless, slightly acidic with a pH of 6.8 viscid fluid consisting of secretion from the parotid, sublingual and submandibular salivary glands and the mucous glands of the oral cavity. According to Tenkate's Book of Oral Histology, saliva is a complex fluid produced by the salivary gland, the most important function of which is to maintain the well-being of the oral cavity. The composition of saliva. The saliva produced by each major salivary gland differs in amount and composition. The parotid gland secretes a watery saliva rich in enzymes such as amylase and protein. Saliva of submandibular gland, in addition to the components already listed, contains high glycosylate substances called as mucin. Sublingual gland produces viscous saliva, also rich in mucil. Oral fluid, which is referred to as a mixed or a whole saliva, includes the secretion of the major glands, the minor glands, desquamated oral epithelial cells, microorganisms and their products, food debris, serum components and inflammatory cells. Composition of saliva. Saliva has water 99% and minerals 0.5%. In minerals, there are organic substances and inorganic substances. Under organic substances, there are enzymes and other organic substances. Enzymes include amylase, maltase, lingual lipase, lysosomes, carbonic anhydrase, and calcarein. Other substances include protein, mucin or albumin, blood group antigen, free amino acid, non-protein nitrogenous substances, urea, uric acid, and creatinine. Inorganic substances include sodium, calcium, potassium, bicarbonate, bromide, chloride, fluoride, phosphate. Others are oxygen, carbon dioxide, and nitrogen. Formation and secretion of saliva. The first stage, the formation and secretion of saliva has two stages. The first stage, cells of the secretory end piece and intercalated duct produces primary saliva, which is an isotonic fluid containing most of the organic components and all of the water that are secreted by the salivary glands. In the second stage, the primary saliva is modified as it passes through the striated and excreted duct, mainly by resorption and secretion of the electrolyte. The final saliva that reaches the oral cavity is hypotonic. The ducts. The ductal system of the salivary gland is a varied network of the tubules that progressively increases the diameter beginning at the secretory end piece and extending to the oral cavity. The three classes of ducts are intercalated, striated, and excretory, each with differing structures and function. The ductal system, connected for the passage of saliva, it actively participates in the production and modification of saliva. Intercalated duct. Primary saliva passes through the intercalated duct. The first cell of the intercalated duct are directly adjacent to the secretory cells of the end piece, and the lumen of the end piece is continuous with the lumen of the intercalated duct. The intercalated duct are lined by a simple cuboidal epithelial and myoepithelial cell bodies, and their processes typically are located along the basal surface of the duct. The overall diameter of the intercalated duct is smaller than that of the end piece, and their lumen are larger than those of the end piece. Several duct draining individuals end piece join to form a large intercalated duct, and these major join again before emptying into the striated duct. The length of the intercalated duct is different. Major and minor salivary gland varies. The intercalated duct cells have centrally placed nuclei and a small amount of cytoplasm containing some rough endoplasmic and small Golgi complexes. Intercalated ducts often are difficult to identify in routine histological sections. The properties. The intercalated ducts contribute macromolecular components which are stored in their secretory granules to the saliva. These components include lysosome and lactoferrin. Other currently unknown components probably also are secreted by these cells. A portion of the fluid component of the primary saliva likely is added in the intercalated duct region. Undifferentiated cells thought to represent salivary gland stem cells, are believed to be present in the intercalated duct. These cells may proliferate and undergo differentiation to replace damaged or drying cells in the end piece and striated ducts. Striated duct. It receives the primary saliva from the intercalated duct, constitutes the large portion of the ductal system. These ducts are the main duct components located within the lobules of the gland, which is intralobular. Striated duct cells are columnar with a centrally placed nucleus and pale acidophilic cytoplasm. This well-presented tissue, faintly radially oriented lines of striated may be observed in the basal cytoplasm of the ducts. The overall diameter of the duct is greater than that of the secretory endpiece, and the lumen is larger than those of the secretory endpiece and intercalated ducts. Basal cell encloses the striated ducts and the capillary flexus is present in the surrounding connective tissue. The properties, the structure of the duct cells reflects an important function of these cells, which is the modification of the primary saliva by reabsorption secretion of electrolytes. The basal secretion, striation of the duct cells, which observed by the electron microscopy, results from the presence of numerous elongated mitochondria in narrow cytoplasmic partitions, separated by high, enfolded, and interdigitated basolateral cell membranes. The apical cytoplasm may contain small secretory granules and electrolucent vesicles. The granules contains calcarein and perhaps other secretory proteins. The presence of vesicles suggests that the cells may participate in endocytosis and substances from the lumen. The duct cells also contain numerous lysosomes and peroxisomes, and deposits of glycogen commonly are present in the perinuclear cytoplasm. Adjacent cells are joined by well-developed tight junctions and junction complexes, but lack gap junctions. The excretory duct is located in the connective tissue septa between the lobules of the gland. Therefore, it is called as the interlobular or extralobular. These ducts are larger in diameter than the striated ducts and typically have the pseudostratified epithelium with columnar cells extending from the basal lamina to the ductal lumen and small basal cells that sit on the basal lamina but do not reach the lumen. The epithelium of the main excretory duct may become stratified near the oral opening. As the duct increases in size, the number of mitochondria and the extent of infold of the basolateral membrane decreases. Basal cells have numerous bundles of intermediate filaments and are attached to the basal lamina by prominent hemidesmosomes. This is the picture which shows the histological picture which shows the intercalated duct, the secretory acini, the excretory duct, and the arteries. This is an image which differentiates between the intercalated duct, the striated duct, excretory duct, and the acinus. Basic mechanism of secretion of granular cells. Macromolecular components. Like other cells that are specialized for the synthesis and regulated secretion of proteins and glycoproteins, the cells of the secretory endpiece have abundant rough endoplasmic reticulum and large Golgi complexes, and they store their products in the membrane-bound granules in the apical cytoplasm. Secretory proteins are synthesized by ribosome attached to the cystine and the endoplasmic reticulum and translocated to the lumen of the endoplasmic reticulum. The proteins are transferred to the Golgi complex where they undergo further modification followed by the condensation and packing into the secretory granules. The secretory granules are stored in the apical cytoplasm until the cell receives an appropriate secretory stimulus. The granule membranes fuses with the cell membrane at the apical surface and their content are released into the lumen by the process of exostosis. In salivary gland, the sympathetic neurotransmitters, norepinephrine, usually is an effective stimulus of exostosis. Norepinephrine binds to beta-adrenergic receptors on the cell surface that eventually leads to the granule exocytosis. After release of the granule content, the granules membrane is internalized by the cell as small vesicles, which may be recycled and degraded. The formation of saliva. The secretory acinus product, the primary saliva, which is isotonic, which an ionic composition resembles that of a plasma. In the duct system, the primary saliva is then modified by selective reabsorption of sodium and calcium without water and secretion of potassium Salivary secretion is of two stages: initial formation stage and modification stage. Initial formation stage involves acinars to secrete primary secretion that contains and mucus in a solution of ions similar in plasma. Modification stage is when the primary secretion flows through the duct and the ionic composition of saliva is modified. Duct modification of saliva. In addition to converting saliva from the secretory endpiece to the oral cavity, an important function of the striated and the excreted duct is the modification of the primary saliva produced by the endpiece and intercalated duct, occurring principally through the reabsorption and secretion of electrolytes. The lumen and basolateral membranes have abundant transporters that functions to produce a net reabsorption of sodium and calcium, resulting in the formation of hypotonic final saliva. The ducts also secrete potassium. The final electrolyte composition of saliva varies depending on the salivary flow rate. At high flow rate, saliva is in contact with the ductal epithelium for a shorter time. The sodium and the calcium concentration raises and the potassium concentration decreases. At the low flow rate, the electrolyte concentration changes in the opposite direction. The modification stage. In the next step, the composition of primary saliva is modified in the duct system. The interlobular duct reabsorbs sodium and calcium, excluding water, and makes the final saliva hypotonic. The properties of saliva: volume is 1 to 1 liter. The pH is 6.3 to 6.85. The specific gravity is 1 to 1.012. And the tonicity is hypertonic. The functions of saliva: it has a multifunctional property, and the main functions are buffering, digestion, mineralization, lubrication, and viscosity, viscoelasticity, tissue coating, antifungal property, and antiviral property, and antibacterial property. Conclusion: an understanding of the anatomy, histology, and physiology of the salivary gland is essential for good clinical practice. In all aspects of the clinical practice, salivary gland and saliva plays an important role. Saliva regulates the oral environment and has widespread distribution of salivary glands in the oral cavity. Hence, there is a great impact of salivary gland pathology on clinical practice in dentistry. Therefore, a complete knowledge of the salivary gland and its physiology and pathology is necessary in treating patients with salivary gland disorders. Thank you.